We're still doing the Chapter 20 lectures, and this is where we left off on slide number 35. Pull it up. Okay, and we were looking at the structure of a typical bacterium, or really just any type of prokaryote, so it could be an archaea or a bacterial cell. And we covered that in the very center there is a very long circular chromosome that's all wound up because it is so long, but it resides, but it's just a single chromosome. That's the point. It's a single circular chromosome, whereas like with humans, we have 46 chromosomes that are strands. They are linear chromosomes instead of circular. So the chromosome is all bundled up and wound up real tight in a region called the nucleoid. And then we have in the cytoplasm, we also have the ribosomes, which make proteins. Then we have a layer surrounding the cytoplasm called the plasma membrane. Outside of the plasma membrane is the cell wall. Outside of the cell wall is a structure called the capsule, composed of the glycocalyx. Sometimes there are little hairs on the outside called fimbriae, and they help with attachment. Sometimes there is a structure called a conjugation pilus that helps with injecting DNA into another cell. Flagella are for movement. And I think that covers it. Okay, so let's move on to the next page. Reproduction in prokaryotes is typically asexual reproduction, and it is called binary fission, which literally means to split in half. So prokaryotes reproduce asexually. Sometimes they can reproduce in as fast as 12 minutes. One prokaryotic cell can divide into two as quickly as 12 minutes. Sometimes it takes longer. One of the ways that they get, because they don't reproduce sexually, one of the ways that they get genetic variation is through mutating. They do have a faster mutation rate than eukaryotic cells. Prokaryotic cells are haploid. That means they only have a single chromosome number. They don't have a double copy of the chromosomes. And some bacteria form endospores that enable them to be resistant when conditions are not favorable for growth. So that enables them to go dormant until conditions are better. This is an image of binary fission. It shows you the nucleoid region of the parent cell. It shows that the single linear chromosome, I'm sorry, I didn't mean to say linear. The single circular chromosome, which is shown in red in the nucleoid region, has replicated. So there's one here, and when that cell divides in half, each new bacterial cell is going to have a copy of it. Now, there are other ways besides mutation to get genetic variation. So there are ways for bacterial cells to undergo genetic recombination, or the recombination of genes, so you can get more variety, more genetic variety. One way is called conjugation, and this occurs through the conjugation pilus. And this is when DNA is transmitted from a donor cell to a recipient cell. Both the donor and the recipient cell are bacterial cells, okay? And that conjugation pilus is used to transfer the DNA from the donor to the recipient. So donor DNA to recipient DNA through the conjugation pilus. Then we have transformation, which we learned about and was discovered by Frederick Griffith. Remember that from Bio 111. This is when a bacterial cell picks up DNA from other bacterial cells, even if they are dead. So they pick up free pieces of DNA from their environment, basically. And transduction is when DNA is transmitted into a bacterial cell from a bacteriophage. which is a virus that infects bacteria. So the bacteriophage or virus serves as a vector. So transduction is when bacterial DNA is exchanged using a virus as a vector. Transformation is when bacteria pick up DNA from their environment, often from other dead bacterial cells. And then conjugation is when the DNA is transmitted from donor to recipient, and both donor and recipient are bacterial cells. Okay, now we're on section 20.3, which is specifically on the domain bacteria. I almost said the kingdom. It's the domain bacteria. Remember, there are 3 domains of living things. 3 total domains: bacteria, archaea, and eukarya. So the bacteria are the domain that we're going to focus on now. They are the more common type of prokaryote. The cell wall of bacteria contains a chemical called peptidoglycan. We make use of that when we stain them using a procedure called the gram stain, and we'll do this in lab, so it's good that we're going over it also in lecture. The gram stain procedure enables us to identify bacteria that are either called gram positive or gram negative, and that just helps us to narrow down the type of bacteria that we are observing. If the bacteria are gram positive, they will retain a purple dye in their peptidoglycan layer, and their peptidoglycan layer is much thicker. So it enables them to retain a purple dye, and they appear purple under the microscope. The gram-negative bacteria have a much thinner layer of peptidoglycan, so they actually appear, it says here that they appear red. They actually appear pink under the microscope. That red shows up as a very light red, and it really looks pink. Because their peptidoglycan layer is very thin, we have to use a different stain with them, and that stain makes them show up, very light red or pink. Gram-negative bacteria have a second plasma membrane which blocks a lot of antibiotics, and that makes the infections difficult to treat. So a lot of our infectious diseases are gram-negative bacteria. And this shows you the gram stain procedure. It shows you how you will flood. You have a sample of bacteria on your slide. You'll flood it with purple stain. Then you'll flood it with iodine, and the iodine will cause the purple stain to lock into the cell wall. And then you'll flood it with a decolorizer, which will allow the gram-positive bacteria to retain the stain, but it will wash it off of the gram-negative bacteria. Then you will flood the slide again with a red dye called safranine, and that will adhere to the gram-negative cells. So this is really showing you the procedure. The purple dye is called crystal violet, so you'll flood it with crystal violet, then iodine, then a decolorizer, which will remove that stain from the gram-negative bacteria. So now on the slide, if you were to look under the microscope, you could see the gram-positive bacteria, but not the gram-negative. So the fourth step is to stain the gram-negative bacteria with the red stain, and then this is what you would see under the microscope. The gram-positive bacteria would appear purple, and the gram-negative bacteria would appear pink. Bacteria can, after we decide if they're gram-positive or gram-negative, we can also look at their shape. If they have a spiral shape, then we call them spirili. They have a rod shape. An individual rod-shaped bacterium is called a bacillus. Plural is bacilli. If they have a round shape, singular is coccus and plural is cocci. And here you can see under the microscope, you can see a spirillum, you can see a bacillus, and you can see a chain of coccus or cocci bacteria. And that shape will help us to determine, that plus the gram stain will help us to identify the bacteria. Now, We talked about viruses. Viruses don't have a metabolism at all. So, but we did say that they were obligate intracellular parasites. So we're going to use that term obligate again here for bacteria, but we're referring to their oxygen requirements. If they are obligate aerobes, that means they cannot grow unless oxygen is present. They are, in other words, they're obligated to grow in the presence of oxygen. So if they're obligate aerobes, they must have oxygen. We are obligate aerobes. If they are obligate anaerobes, they can't grow in the presence of oxygen. So if oxygen is present, they die. Oxygen actually kills them. And examples are the bacteria that cause botulism, called Clostridium botulinum, the bacterium that causes gas gangrene, and the bacterium that causes tetanus, which is another Clostridium tetani. So these are obligate anaerobes. That means oxygen will kill them. If they are facultative anaerobes, that means that they can grow either in the presence of oxygen or in the absence. Some bacteria make their own food, so we call them autotrophs. If they are photoautotrophs, they don't necessarily photosynthesize, but some of them do, but some don't. Photoautotrophs use sunlight or solar energy to convert carbon dioxide into food in the form of carbohydrates. If they're photosynthetic and oxygenic, then we call them cyanobacteria. But if they're photosynthetic and anoxygenic, meaning that they use solar energy to convert carbon dioxide into food, but they don't use oxygen to do it. Okay? If they're anoxygenic, then they are either green sulfur bacteria or purple bacteria that live in oxygen-poor conditions. So the oxygenic bacteria, I need to write this in here. I hope I can, because I have trouble doing that sometimes. But they are called cyanobacteria. Sorry. C-Y-A-N-O. And then in your mind, you can just put bacteria on the end of that word. Cyanobacteria are the oxygenic photosynthetic bacteria. Okay, that means that they use oxygen. I'm sorry, I keep saying use oxygen. I don't mean that. I mean that they use carbon dioxide to make carbohydrates, and they release oxygen. They release oxygen as a byproduct. Okay, so that's the cyanobacteria that carry out photosynthesis and release oxygen as a byproduct. But green sulfur bacteria and some of the purple bacteria can also carry out photosynthesis. They just do not release oxygen. Then we have chemoautotrophs, and these use the energy from chemicals to convert carbon dioxide to carbohydrate. So the energy comes from inorganic compounds instead of sunlight. But they still take carbon dioxide and convert it into carbohydrate. And a lot of these live in deep sea vents, 2.5 kilometers below sea level. So we have a table here that shows you the photoautotrophic bacteria, the ones that carry out photosynthesis but don't produce oxygen are anoxygenic. The ones that carry out photosynthesis and produce oxygen are the cyanobacteria. And then we have chemoheterotrophs, like humans. Chemoheterotrophs all eat organic nutrients. They consume organic nutrients. And so they get their food and they get their energy from the breakdown of those organic nutrients. So they have to consume them. They don't make their own food. Saprotrophs are decomposers. So these are bacteria that decompose organic molecules. And then we have symbiotic